Yay. Good morning, everybody. I hope you had a lovely spring break. Thank you for being patient with me this morning. And it looks like maybe some people did not get the memo because there's only 17 people in the class, but that's okay. I'll post the recording afterward. Yeah, I'm still adjusting to the whole daylight savings time. I hate spring forward, fall back, spring forward, fall back. I hate that. We should just spring forward and then that's it. Call it a day. There's no more reason for daylight savings time. And as many of you probably know, a lot of countries don't even do the whole daylight savings time thing. So I am not a fan. I'm still adjusting. Yes. So in other words, it's like 9:20 to me right now. Still getting used to that. Cool. So let me find the PowerPoint.
That's the wall.
Hey, hello. Make sure you guys please mute. All right, this is the PowerPoint. So let's pull this up. Share screen. Go to the Cycads, finish them up. Yes, hopefully you guys can see the PowerPoint. Let me pull up, make it big. Let's see, do I remember how to do this? I'm kidding. Make it big, let's see. Okay, move this. Yeah, so last time we did not finish the Cycads before break. All right, please mute. Thank you. We didn't finish the Cycads before break. So what that means is that we, yes, Brittany is asking in the chat box, is this lecture 13? We did not finish lecture 13 prior to the break, right? We ran out of time. And so we're gonna finish the rest of lecture 13 today, right? So Cycads is lecture 13. We're finishing that today. And that means that the Cycad assignment is going to be postponed. So initially it was due today, and then I had postponed it to this coming Thursday. We'll see how we do with the material today. I think we'll be able to finish the Cycads today. And if that's the case, then the next assignment will be due a week from today. So stay tuned for that. I will make a decision after class, depending on how much we get through the material. And then I will post on Blackboard to let you guys know when the new assignment due date is. Hopefully you guys have been taking pictures of your trees. Some of you have emailed me questions. Thank you. If anyone needs any clarification about the phenology project or wants to show me some pictures thus far, that's fine. Send them my way and I can give you feedback. Okay. All right, so let's see, we need to finish this. So what do they say? It's like previously on Cycads. No. We kind of went through this whole week. Last time, what we kind of finished up is learning about this concept of, here, I'll go back. Secondary growth, right? Like we learned about secondary growth. These things might look a little familiar or not because it was before spring break and you maybe were in that class, you were tuned out. Well, tune in now, please tune in now. But the point is, the last time we talked about secondary growth, which was a new concept to us, in this class. And the reason why we spent so much time talking about secondary growth last time is because all the other plants, pretty much all the other plants we're going to look at going forward are going to have not only primary growth, but also secondary growth. So that's why we spent so much time talking about it last time. The last time we went through the structures and the tissues that make up that secondary growth. But the main takeaway of all that discussion was that Cycads do have secondary growth, but it is limited. It is limited. And what I mean by limited is that, yes, Cycads can, remember, secondary growth is to increase in width, and primary growth is to increase in length. So yes, cycads do make secondary growth, but not that much, the tissues are not as robust as they will be in other species that we look at. Okay. Not going to go through the seal again, not going to go through this again, but just to get you kind of oriented of where we left off last time. It's like a long discussion about secondary growth. But today what we need to do is finish cycads. And to do that, we need to talk about cycad reproduction. So, cycads, right? They are vascular plants. They are seed plants. They are seed plants. If they're seed plants, that means that they also have had ovules, because you have to have an ovule before you can have a seed, right? So let's talk about this cycad reproduction. So here we had a whole lecture about the ovule, right? So we've come across some of these terms before. For example, megasporangium, we know what a megasporangium is. And then when we talked about the ovule the first time, we learned these concepts, these words, micropyle, which is a hole in the ovule that allows the sperm to enter. Let's see, right? Micropyle, look, there's the hole right there. We learned about the integument, which is a layer of tissue that surrounds the megasporangium and thus providing more protection for not only the megaspore, which grows into the female gametophyte, but then later the integument is going to undergo chemical changes and turn into the seed coat, which we'll talk about more in another time. And we learned about this concept of functional megaspore, right? Because in the other plants that we've talked about so far that have had megaspores and microspores, right? For example, Selaginella. In those plants, each megasporangium had 4 megaspores inside that would all get released. But now in this seed plants and in the cycad, there's really only one functional megaspore. And we talked about how that megaspore comes about in the ovule lecture. So go back to that if that's kind of fuzzy. Point of this slide is that, yep. Got a question there? No, just unmuting, okay. Yeah, so we're talking about cycad reproduction. No problem, Nicole. Talking about cycad reproduction. And so one player in cycad reproduction is ovules, right? Because we're in the seed plants. Inside every ovule is a megasporangium, and inside of every megasporangium is a functional megaspore, right? We talked about that already. You also have microsporangia. So you have megasporangia inside of ovules, and you have microsporangia, right? And the microsporangia, just like we're used to, produce microspores. And these things on the right are lots and lots and lots of microsporangia, right? Look, they almost look like little Pac-Men. I don't know if you guys know Pac-Man. I hope so. Pac-Man, he's like this little like yellow, like mom, eats things, gets kind of like chased by these, I think they're ghosts. Catherine says they look like pasta, kind of. But they look, basically those little yellow things are each of those a microsporangia, and each of those microsporangia are going to release lots of microspores, right? So when you're looking at the slides later, there is one microsporangium right there, okay? So just like we're already used to, we have special leaves that carry these structures. So ovules, which have megasporangia inside, and microsporangia are both attached to leaves Ovules are on leaves called a Megasporophyll. Microsporangia are on leaves called a Microsporophyll. And just so that you kind of get back to speed on these things, right? Whenever you see these long words, what do I recommend that you do? You break them down, right? You break them down. Fill. What does fill mean? What does fill mean? Like my friend Phil, hey Phil, like no. What does fill mean? Yes, okay, so people, Diksha and Taser are writing in the chat box, leaves, right? Yes, so leaf. So fill means leaf. And then if you're a fill, if you're a sporophyll, what does the leaf have on it? What does the leaf have on it? Here's a clue. If you're a sporophyll, it means that you're a leaf that carries a sporangia on it. Sporangia, not simply spores. So a few people are writing in the chat box spores. Not simply spores, because spores aren't just out and about, right? Spores are inside of a structure, in this case, or in every case, a sporangia. So if you're a sporophyll, it means you are a leaf that carries a sporangia. And if you are a microsporophyll, that means that you are a leaf that carries microsporangia. And if you're a megasporophyll, that means you are a leaf that carries megasporangia. Sorry, there's like a loud truck outside. I don't know if you guys can hear that. Sorry. Okay. Yes. So cycads have ovules. Cycads have microsporangia. And both of those things are on leaves called a megasporophyll and microsporophyll, respectively. Loud truck. So in cycads, remember, we're talking cycads here. In cycads, megasporophylls are sometimes arranged in a strobulus. Sometimes. Other times not. It depends on what species you're talking about. But in cycads, microsporophylls are always going to be arranged in a strobulus. And this is a good time to point out, like when we go forward in the seed plants, next plants we're going to talk about after cycads are, sorry, loud truck. After we talk about cycads, then we're going to talk about ginkgo, which is a seed shedding plant, and then eventually conifers. And I should point out, in ginkgo and conifers as well, the microsporophylls are always arranged in a strobulus. So this is a trend, helps us remember. Helps us to remember if we like see these trends. So megasporophylls sometimes arranged in a strobulus, microsporophylls always arranged in a strobulus. And you're like, Rebecca, I just had spring break. I'm all refreshed. What the heck is a strobulus? I don't want to, what is a strobulus, Rebecca? Okay, remember, we've come across strobulus lots of times already, but just as a reminder, a strobulus is a structure, and it consists of a stem, leaves on the stem, and on those leaves, sporangia. And that's the same structure that we saw in the lycopods when we first came across this concept of a strobulus, right? So strobulus always consists of these things. In the cycads, the strobili look maybe a little different than we're used to seeing. So these pictures below are examples of cycad strobili. Almost kind of look like, some of them almost kind of look like a pineapple or something like that, right? To me, they look very prehistoric, like, and remember, I said cycads are plants the dinosaurs ate, right? And those old pictures of like dinosaurs eating a little cycad leaf, right? So it kind of looks Jurassic Parky in a way. What's cool though is that Cycads strobili are just very, very diverse. They come in all different colors, all different sizes, all different shapes. I think somebody, if they wanted to, could spend a whole career studying Cycads strobili just because they are very, very different and beautiful. I'm a huge fan. So these are some examples shown here. Okay. All right. More terms that we have to get reminded on. So cycads are strictly dioecious, which means that they have sex organs onto separate plants. Female sex organs on some plants, male sex organs on other plants. The female plants bear only female sporophylls, right? So megasporophylls. And remember, I said the megasporophylls are sometimes, nope, what is, oh, spam call. Remember I said on the previous slide, the megasporophylls are sometimes arranged in a strobulus. So on the female plants, depending on the species, you'll either have the megasporophylls arranged in a strobulus or in what's called a crown of fertile leaves. And I will show you what that looks like on the upcoming slide. So this is a picture, or a drawing rather, of two different female plants from two different species. And let's do yellow, I guess. This here is like what we're used to, a strobulus with, you know, a stem, leaves, and the leaves have sporangia on them. Whereas this species here has what's called a crown of fertile leaves. Those fertile leaves in that crown are megasporophylls. So the leaves on the strobulus are megasporophylls. The leaves in the crown are megasporophylls. Depends on the species. Here is an example of a species that has the megasporophylls arranged in a strobili. And note, these are not cones. I think the book even calls them cones. They are not cones. Even Dr. Casper would make a big point about this, because a cone is a different structure. We'll learn the parts of a cone later. But suffice it to say, a strobili is a stem with leaves on it. A cone is not a stem with leaves on it. Let's just put it that way. But here, so all three of these are strobili, different versions of strobili, right? So this whole thing here is one female strobulus, right? So a stem with megasporophylls that have megasporangia on them, right? Ovules with megasporangia in them. Here's a drawing of the same thing. And for what it's worth, the megasporophylls don't really look like leaves that we're used to. So these kind of like, they almost look like those crackers that you put in soup. What are those called? Oyster crackers? Each of those things is a leaf, right? So here's another one. Each of these things is a leaf or a megasporophyll, right? Here, here's another one on this one. And yeah, like they don't look like green leaves that we're used to seeing, right? But remember, even in the lycopods, when we talked about strobili, the leaves on those strobili also weren't green, meaning not photosynthetic, right? So we're kind of used to this. And on each of these leaves will be one, or it depends on the species, but on each of these leaves will be ovules. So those are the red things kind of poking out here. This is an ovule here. On this species on the right, it's a little difficult to tell because the megasporophylls are red and the ovules also happen to be red. But trust me, there's ovules there. So these are strobili, not cones, not cones. So this is an example of megasporophylls that are arranged in strobili. Now we'll look at a species where the Megasporophylls are arranged in what's called a crown, right? A crown. So this is our crown of fertile leaves on the left. Now here, Megasporophylls look a little bit more like a leaf, right? Like this thing looks more like a leaf, right? That looks like a leaf. More like a leaf than the little soup crackers that we saw on the previous slide, right? And this whole structure here is the crown. This is a crown of fertile leaves. This is a crown of fertile leaves. Fertile leaves meaning they have an ovule on them, or ovules depending on the species. And inside the ovules is the megasporangia. Okay, so those are the females. Oops. Just so when you're looking at it later by yourself, you have some orientation here. The Megasporophylls in Cycads also are very diverse and all different shapes and sizes and colors. So not only are the Strobili diverse in that way, but the Megasporophylls themselves are also diverse in that way. So all these here are examples of Megasporophylls. Trust me. So like this whole thing is a Megasporophyll, this whole thing is a Megasporophyll, and each of these things, believe it or not, is also a Megasporophyll. And as I said in the previous slides when I was talking about this, the number of ovules on each Megasporophyll will depend on the species and depend on the shape of the Megasporophyll itself. So on this Megasporophyll to the left, this has 6 ovules. 3, 4, 5, 6, right? The one next to it has 5, and the 3 on the right each bear 2 ovules, right? There's one, there's one. The leaves on the left-hand side look more leaf-like, maybe not the one here, right? Maybe not this one, fine, but they look more leaf-like than the ones on the right. And that's because the leaves on the left, these two, are examples of leaves that will be in a crown of fertile leaves, whereas these 3 will be in strobili, Okay. But the point is a lot of diversity in Megasporophyll shape and different numbers of ovules depending on the species of cycad. And you have this for when you're reviewing it on your own. Usually, the Megasporophylls that are arranged in a Strobilus have 2 ovules, just because those Megasporophylls are very small, so they can only really hold on to 2 ovules, whereas the Megasporophylls of a fertile crown tend to be a lot bigger and therefore can bear more ovules. Right, and this is what I just said. Okay, so those were the female structures in cycad reproduction. Now we're going to talk about the male structures. So remember, I said in the female plants, sometimes in the female plants, the Megasporophylls are arranged in a strobili, sometimes they're not, and they're in a crown of fertile leaves. But on the male plants, the Microsporophylls are always arranged in a Strobilus. Always arranged in a Strobilus. And we know that the Strobilus consists of stem, leaves, and microsporangia. And in the case of the cycads, the sporangia or the microsporangia are on the bottom, remember axial? For those of you going to medical school or dental school, this is a good prefix to remember. Ab means away. Here, I'll write it in the chat box. Ab equals away from, or sorry, ab is bottom, whereas ad is top or dorsal. But abaxial surface of the leaf is the bottom side of the leaf, which is what so we've seen, this is a good time to compare, right? We've seen sporangia on both the top and bottom of leaves so far in this course. For example, in the lycopods, right, Lycopodium and Selaginella, the microsporangia, or the sporangia were always on the top of the leaves. But in the ferns, the microsporangia are always on the bottom of the leaves. And in the cycads, the microsporangia are also on the bottom of the leaves. Here is examples of what that looks like. So on the left, you see an example of a male Strobilus. In the middle is picture of a male Strobilus. So this whole thing is the male Strobilus. And it consists of many microsporophylls on this Strobilus. And if we were to pull off one, pluck off one microsporophyll and look at it closely, here's the whole microsporophyll, and each of these little things, each of these little dots on the bottom side of that leaf are the microsporangia. And remember, those microsporangia are those things kind of look like little Pac-Mans. On the right here, this is a cross section. So let's say we did this. We're going to do a cross section through a male Strobilus and look at it. So this is what that is on the right. Each of these things here are the Microsporophylls. And all those little dots on the bottom side of the Microsporophylls are those microsporangia, right? Those little Pac-Mans. So just so we get oriented, right? What is a Strobilus? It has a stem, right? Here's your stem. Here's your stem. This is your stem because this is a cross section. Leaves, which are in this case Microsporophylls, which are all of those things that you see here, all these little things. And in the cross section, here's one. And in the cross section, there's one. And I pulled it off of the Strobilus, there's another Microsporophyll. And microsporangia, which in the drawing here are all these little dots. And in the picture on the right are these little Pac-Man looking circles. Okay, those are the male Strobili. Because remember, cycads, dioecious, male and female plants, male and female sporophyte plants, male and female sporophyte plants, which I know is a little confusing. I should say, right. Right, because, okay, confusing. I don't want to get you guys confused. Just know that there are male and female cycads, and that the male plants are going to ultimately make sperm, and the female plants are ultimately going to make eggs, right? It's a little more complicated now because the gametophyte generation is so reduced, it's kind of part of the sporophyte plant. We'll talk more about that in the slides coming up. Okay, so there's your Strobilus. And I think I circled, yeah, there you go. All right, and that's I knew that this is where we were getting to. All right, cycad reproduction. As I said, the male gametophyte plant, so all the gametophyte plants now are reduced, right? Because in the plant kingdom, we see over time a reduction in size of the gametophyte plants, right? That is a trend, right? That is a core concept of this class. is that gametophyte plants over evolutionary time get smaller and smaller and smaller, more and more and more reduced. And since we're in the seed plants now, the gametophyte plants are even going to be more reduced than what we saw in the spore shedding plants. So you may remember that I said back in the day, that when this heterosporous plants, like when we go from homospory to heterospory, it's a major shift. And the reason why it's so significant is because it is the precursor to the seed shedding plants. So now we're in the seed shedding plants. And the gametophyte is even more reduced, So this slide tells you to remember or recall that in Selaginella, the male gametophyte plant consisted of a single antheridium inside of a microspore, right? So this is a slide from the Selaginella lecture. Here's one microspore. Let's try that again. Here's one microspore. If you were to look inside of that microspore, there would be just one antheridium, right? Remember testicles in space? You're like, I'll never forget that. Thanks a lot, Rebecca. But yes. So that's what we saw in Selaginella, right? Which is a spore shedding plant, right? See, there's only one antheridium inside. And that's what the entire male gametophyte is, right? A single antheridium inside of a microspore. That's what we saw in the past. Right. Oh, here we go. Pollen grain. So in the past, we saw the male gametophyte plant consisted of a single antheridium inside of a microspore. But now we're in the seed plants. And we have a huge evolutionary leap, which is the pollen grain. So you may remember from this whole, we know, here, let me just like get out of this for a second. Take a break from that. We know that inside of any sporangia, right, there are sporocytes, right? So a sporangia is diploid, and inside of the sporangia, of any sporangia, are diploid cells that are going to undergo meiosis and cytokinesis to produce haploid spores for any sporangia that you ever talk about, right? We know that already. If you're talking about megasporangia and microsporangia, the megasporangia and microsporangia are going to have inside of them megasporocytes or microsporocytes that are diploid, that are going to undergo meiosis and cytokinesis to produce haploid megaspores and haploid microspores, right? Same thing, it's just that now there's two types of spores, right? Same thing, but two types of spores. So let's go back to this now. Share the screen. Here it is. Share. Cool. Right. So I just said all that stuff, right? About what's happening inside of sporangia. Well, now we're in the seed plants. We have microsporangia, we have megasporangia. This is what I just said. Inside the microsporangia are these microsporocytes that are diploid. They're going to undergo meiosis and cytokinesis to produce tetrad of microspores, right? Tetrad of microspores. And that tetrad of microspores is ultimately going to lead us to this huge evolutionary leap, the pollen grain. And I will show you how right now. Yes. Inside the microsporangia, the microspores undergo, oh yes, so, right. So tetrad of microspores is the product of meiosis and cytokinesis. And then each of these microspores, each of them are going to undergo mitosis or grow. Did I like cut out there at all? If I do cut out, just write in the chat box. Because if need be, I could plug into the modem. Here, let's see. Okay, thank you, Benjamin. But if I do start to like lag, let me know and I could plug into the box. Anyways, so each of those microspores is gonna then undergo mitosis to grow. When you hear mitosis, you think grow into pollen grains. So this is what I just said. This is an even smaller, more reduced male gametophyte plant, aka the pollen grain. What is a pollen grain? What does the pollen grain consist of? It consists of a few cells, only a few cells now. That's how reduced we are. We were, in the Selaginella, we were a whole antheridium inside of a microspore. Now we're just a few cells. The male gametophyte plant is only a few cells. These cells are the prothalial cell, which we really don't know what that does. So we think maybe it's leftover. Which is another way of saying that is vestigial. I don't know if you guys have heard of like vestigial organs, right? Like I think the appendix in humans is a vestigial, well, I mean, it has uses, but like basically an organ that's there or something that's there, in this case, the cell, where we're really not sure what it does. It may be leftover. Like, for example, whales have hip bones, right? Whales have hip bones. Whales don't have legs. But they have hip bones, so vestigial. So prothalial cell, pollen tube cell, which as the name tells you, will grow into a pollen tube, and then a generative cell, which is going to generate sperm. Motile means swimming, just so you know. To motile means swimming. To swimming sperm, okay. Here is a sperm. Here's a sperm. And so just like we've seen in previous plants that we've talked about, the sperms swim. However, one difference now is that they don't swim through water. They swim through other things, which we'll talk about. But they still swim. Part of the way you can tell that this sperm must swim is because it has all the cilia. Look at all these little, they're like tails. All these little tails that will kind of wag and allow it to swim. Right, they don't swim in water anymore, but they're gonna swim down the pollen tube. All right, so we're gonna go through this process, and I think we'll be okay to finish up Cycads today. So here, what we're going to look at right now is the pollen grain and its parts. So here is a microspore, right? That would have been the result of meiosis and cytokinesis inside of a microsporangia, right? Microspore, which is haploid. Microspore is going to undergo mitosis and cytokinesis, right? Growth. The microspore is going to divide into more cells, right, via mitosis and cytokinesis, and the result of that will be the pollen grain. As I mentioned on the previous slide, pollen grain consists of just a few cells, only a few cells, no antheridium, right? Just a few cells. Those cells are the pollen tube cell, the generative cell, and the prothalial cell. Prothalial cell, we don't really know what it does. So going from the bottom here, this here is the prothalial cell. This here is the generative cell. And those cells are inside of the pollen tube cell. So the whole thing is pollen tube cell. These other cells are inside. Eventually, the pollen tube cell is going to grow into a pollen tube. Here is the pollen tube, allowing this. Oh, and so eventually sorry, I should take a step back. The generative cell generates two sperm, right? That we saw here. Here's a sperm, right? And I said they're going to swim down the pollen tube. The generative, oh, too many boxes on my screen. Generative cell is going to generate two sperm, right? It's this guy here is going to generate two sperm. Pollen tube cell is going to elongate. Here it is, growing into a pollen tube. And that's going to allow the two sperm to swim down. Here they are, swimming down the pollen tube cell. Okay. All right. This is a huge evolutionary leap because now look how reduced the male gametophyte plant is, right? It's only a pollen grain, right? It's only 3 cells. That's a big reduction compared to what we saw in earlier classes, right? When the gametophyte plant was an entire plant. Now we're just 3 cells for the male gametophyte plant. The microsporangia, so those Pac-Man guys, can also be called a pollen sac, because ultimately what gets released from the microsporangia are the pollen grains. So the microspore no longer gets released because the microspore stays inside of the microsporangia and undergoes this process. And undergoes this process. This process, not the one on the bottom. Sorry. This process occurs inside of the microsporangia. Occurs inside the microsporangia. And so that what gets released from the microsporangia is the pollen grain. So we can call the microsporangia a pollen sac. So here you go, one Pac-Man guy, right? Which is one microsporangium, or now we know we can call it a pollen sac because it's going to release pollen grains. All right, so before we wrap up for the day, we have time. We're going to go through alternation of generations in the cycads to kind of get oriented and put together all the things that I just told you about, right? Ovule, pollen grain, and how those two things come together. So that's what we're going to talk about. Remember, cycads are dioecious in that they have male and female plants, right? Separate plants. So here on the left, you have a female sporophyte plant, and obviously it's 2N. And on the right, you have male sporophyte plant, which is also 2N, right? That part's easy enough. On the female gametophyte plant, let's say, so we said on previous slides that some, depending on the species, right? Let me use yellow. Here's our female gametophyte plant on the left. Some species have the Megasporophylls arranged in a Strobilus, right? Some species have Megasporophylls arranged in a fertile crown. For simplicity's sake, let's pretend let's deal with a female sporophyte plant that has her Megasporophylls arranged in a Strobilus. So that's what we're going to go with right now. So here we are. Here's a Strobilus from that female gametophyte female sporophyte plant, right? Here is the Strobilus from the female sporophyte plant. On each Strobilus, right? Strobilus is a stem. Here's the stem, leaves. In this case, they look like those like kind of oyster cracker things, right? Here is a leaf pulled off. Here's a leaf pulled off. And each leaf, in this case, will have 2 ovules. Remember, I said usually when you have the leaves arranged in a Strobilus in the Cycads, each leaf will bear 2 ovules. Don't worry about the number of ovules though. Just know that you have a female Strobilus, which has Megasporophylls that are, all those are diploid obviously, and that each Megasporophyll is going to bear ovules. Note that the ovule is 2N. Note that the ovule is 2N. Okay. This is just to remind you of the top and the bottom side of the leaf. Don't worry so much about that. All right, and then on the male plant, you have male Strobilus, and both Strobilus have the same things: stem, leaf, sporangia. That's both Strobilus have that. But on the male Strobilus, we have Microsporophylls. Those are the leaves, and each Microsporophyll is going to have on it lots of microsporangia, aka pollen sacs, because they're going to ultimately release pollen grains. Top and bottom of the leaf, right? Top and bottom of the leaf. Okay. So let's take, this is our female, so this is our Megasporophyll with ovules. There's one ovule right there. And as we learned in the ovule lecture, the ovule has these parts, right? So inside of the ovule, which is 2N, there is a megasporangia, which is also 2N, containing a megasporocyte, which is also 2N. That megasporocyte is going to undergo meiosis and cytokinesis, the result of which will be one functional megaspore. That functional megaspore is going to undergo mitosis, right? Grow. It's going to grow and divide. And the result will be one female gametophyte plant, which will be living inside of each ovule. That's what's going on there. Notice, again, this trend in evolutionary time. Look at the reduction of these gametophyte plants, right? Big, big reduction. Here's our Microsporophyll. Here is one microsporangia. And if we were to look inside of that microsporangia, we would see microspores. Those microspores would go through, sorry, step back. I jumped ahead because of the picture. Inside of each microsporangium, which is diploid, there are microsporocytes, right? The spore mother cells, which are also diploid. Those spore mother cells are going to undergo meiosis and cytokinesis to produce microspores, which are haploid. There should be a 1N next to that microspore. Here's one microspore up here. That microspore is going to divide and grow via mitosis and cytokinesis into the pollen grain. Here is pollen grain. Pollen grain consists of a few cells and is now our highly reduced male gametophyte plant, right? Because it's only just a few cells, super reduced. Okay, so obviously the ovule has inside of it ultimately, after all these processes, the female gametophyte, right? So we know there's an egg. We know there's an egg. If we have a female gametophyte plant, we know there has to be an egg. And we know that we end up via this process, right, via the pollen grain, with sperm. So, oops. So egg and sperm have to come together some way, They have to come together some way in order to complete in order to go through the alternation of generations. Egg and sperm must come together. So how do they do that? I will show you. All right. This is going to seem a little like, what today, but these things are going to be, a lot of these things are going to repeat in the subsequent classes, so do not worry too much about it today, but I want to get you used to it. And I recommend after this class is over today that you review these slides in anticipation of the Cycad assignment, right? You're going to have to like sit down and kind of review these slides a little bit for this to kind of make sense. But to help you along the way, I've color-coded things on these slides. So notice that as we go forward. All right, so here, this is a cross-section through an ovule. And inside that ovule is that female gametophyte plant, right? We already discussed how that came about. The female gametophyte plant has on it archegonia, right? Here they are. And inside of each archegonia is an egg. So there are 2 total eggs in one female gametophyte plant. Notice, because we're in the seed plants, there's so much more protection for this female gametophyte plant and her eggs, because the female gametophyte plant is inside of the megasporangium, aka nucellus, right? Never gets released. And not only is she inside of this megasporangium, but she's also inside of the integument as well, many layers in this case. So her eggs are very well protected. Here is the microsporangia. So they're going to open and release the pollen grains. So goodbye, pollen grains. See you later. There they go, out on the wind. Bye, bye, bye. Fly away. Pollen grains fly away. Or get brought by insects. And so what happens is the pollen grains get released. I think, let's go, yeah, blue is good. Pollen grains get released into the air. Here is an ovule. Here's an ovule, see, ovule. And the ovule is going to produce this kind of sticky drop, which is called the pollination drop. So this is it coming out here. Actually a little too big. Kind of looks like a ladybug in this picture, but this sticky drop is going to emerge from the ovule through that hole, the micropyle. And when that sticky drop is out, pollen grains that are going by can stick onto that sticky drop, the pollination drop. Here in the drawing, these little black dots are the pollen grains. And when that happens, that's what we call pollination. So when the pollen grain lands on the ovule, right, on the sticky drop of the ovule, this is a process that we call pollination. Pollen grain lands on ovule. This can occur via wind or insects in cycads. So pollen grain lands on ovule. Okay, we still got a little bit of time. Pollen grain lands on ovule, pollination occurs. Eventually that sticky drop, so here, just so you get oriented in this drawing, let's switch colors. Here is, oh, I gotta move this thing. Here we go. Here is that hole, right? So here's that hole, which is called the micropyle. And the sticky drop will have emerged from that hole. And pollen, oops. Oh, come on. And pollen grains can land on the sticky drop. Eventually, the sticky drop, the pollination drop, is going to dry up and go back into the ovule. And when that happens, it's going to bring the pollen grains with it. Micropyle is the hole. As the pollination drop dries up and brings those pollen grains with it, they're going to enter a part of the ovule that's called the pollen chamber, right? So notice pollen chamber is here. I'm going to draw this. Here is the wall of the this is the wall of the megasporangium, sorry. So the pollen chamber is still outside of the megasporangia, still outside, can't get in yet. But that's where it's going to enter first, into that pollen chamber. When it's at the pollen chamber, that's going to allow, so the pollen grain is going to land on the wall of the megasporangia. And it's the pollen tube cell is going to grow. Pollen tube cell is going to grow. So look, just so you know here, look, micropyle. This is where the sticky drop would have come out. It would have dried up and brought the pollen grain into the pollen chamber. When the pollen grain is there, it allows pollen tube cell to grow through the wall of the megasporangia. Here's the pollen tube cell growing, and that's going to allow the two sperm that the pollen grain made to enter the megasporangia. So this is a funny slide, but look here. Just note, look, pollen tube reaches the inside of the megasporangia. That's what I just said. And then the swimming sperm can enter what's known as the archegonial chamber. Again, these terms, pollen chamber, archegonial chamber, micropyle, megasporangium, integument, all those things we're going to see many, many times going into further slides. But the pollen tube cell grew, allowed the sperm to enter this new chamber, where archegonial chamber, so the chamber where the archegonia are located. Each archegonium on that female gametophyte plant has an egg. And so now we can have fertilization occur. Yeah. So when you're reviewing the slides later, you can see where these chambers are. Okay, one sperm. Notice there are how many sperm? 2. And notice there are how many eggs? 2. But both sperm are not going to be successful. And both eggs are not going to get fertilized. Only one sperm is going to be successful, and only one egg is going to get fertilized. So one sperm fuses with one egg. So when sperm and egg fuse, process is called fertilization. We already know that. The other sperm is going to degrade. So it's kind of like a race. It's kind of like a race between the two sperm. And whoever fertilizes their egg first wins, the other sperm loses, degrades, and the other egg goes unfertilized. Which, in terms of evolution, this is pretty wasteful. You don't really need, this is pretty wasteful. Degrade, egg goes unfertilized. That's kind of wasteful. So when we go into the more evolved seed plants, we'll see that this doesn't happen anymore. But for now, cycads are kind of primitive, and the other egg goes unfertilized. And we mentioned in the first part of this lecture on cycads that many cycads are facing extinction, and that's part of that is because it can take months from pollination to fertilization to occur. So pollination, remember, is when pollen grain lands on the ovule. And in the cycads, after that happens, it can take months and months and months and months and months before pollen tube cell can grow into the ovule and the sperm can reach the egg inside. So that's part of the reason why many species are facing extinction. Oh, and I think we're finishing here, but this is one example. Remember how I always made a big deal about, like, you don't always have cytokinesis right away, right? Sometimes you have, so when I would say mitosis and cytokinesis, right? Or meiosis and cytokinesis, because sometimes you can have no cytokinesis occur after mitosis. So that's something to keep in mind when if you're going into medical school, you'll need to know that. So look, sperm and egg fuse. Sperm and egg fuse result is a 2N zygote. 2N zygote. We're used to that. 2N zygote. So here you go. And what's going to happen is that 2N zygote is going to undergo many, many, many mitotic divisions with no cytokinesis in what's called a free nuclear stage. So look, see all these nuclei that are occurring, but they're all occurring inside of one cell. Eventually, the nuclei will get kind of like arranged along the edges of that cell, and then cytokinesis ultimately occurs. So like you can see the little walls, each of the nuclei are now inside of a wall. This process, this kind of thing happens in other organisms as well. For example, fruit flies, this kind of thing happens, free nuclear stage. Then eventually when cytokinesis finally does occur, you end up with the embryo. So this is an embryo. And we were an ovule when we had an egg, but after the egg is fertilized and after you end up with an embryo, we call the whole structure a seed. Again, there's going to be a lot of repetition about this stuff going forward. Let's see, I think, yep, great. We're going to finish up right here. These are parts of the embryo. Don't need to spend a lot of time on those today, but just know there's embryonic leaves, which are called cotyledons. So I'll just draw it quickly. I know we're running out of time. This is an embryo inside of the seed. So I'm going to draw it. That whole thing is embryo. Embryo has embryonic leaves, which are these things. Embryo has an apical meristem, which is here. And there's also one here at the bottom of where the roots will be. And then it has a stem. Just so you know, hypo means below or under. So this is a stem. Oh, sorry, I should use different color. Hypo means below or under. So this is a stem that is hypocotyl or below the cotyledon. So that's this. And there'll be an embryonic root, which is here, coming out of here. We'll talk about embryos a lot in subsequent classes, but just so you get a little oriented. And then eventually what happens is the female gametophyte plant is going to become a food source for the embryo. So the embryo, as it grows, will eat the gametophyte plant, female gametophyte plant, and allow it to grow. This is some seeds. And then eventually the seeds with the embryos inside are going to fall off the tree. The seed will land on the ground and grow into the new sporophyte plant, which is shown on the right. So that's alternation of generations in cycads. We finished it. Yay, we finished it. I really recommend you look at these slides closely. I'm gonna make a decision about when this assignment will be due. Yes, but we covered a lot of new stuff today. Don't feel overwhelmed. I think I'm gonna make the assignment due a week from today, but like I said, I will confirm that via Blackboard. Next time on Thursday, we'll talk about the next member of the seed plants, which is this living fossil tree called Ginkgo biloba, so which is also in the gymnosperm. So if anyone has any questions, I'm happy to answer them now or in a follow-up email. Sorry we ran over a few minutes. Hope you guys have a great rest of your day. And yeah, I will see you on Thursday, and I'll let you know about the assignment. So if anyone has any questions, happy to answer it. Otherwise, have a good one. See you guys next time on Thursday. And I think I have a meeting with a few of you today. So if we do have a meeting, I will send you an email now with a link for the Zoom. Okay. Bye, everyone. Email me if you have any questions. Bye, guys.